we have different systems up here that kind of show us where the light is. So you want to keep your highlights lower than a certain like percentage. So it's a mixture of getting your eyes used to it and understanding where it should be. You don't want to blow things out or else you won't see details. And a lot of those sponges are quite hard because as soon as you have the ROV lights on them, the highlights in the animal actually go through the roof. So you're having to go quite dark, which loses the blacks in the background.
Yeah. Yeah, they're perfectly white. It must be really hard. Anybody who's
and those ROV lights definitely light them up even more.
Anybody who has either a pure white or a pure black dog knows how hard it is to get a nice picture of them.
Well, you know, when you got a sponge in view, why do you want to look at anything in the background anyway?
Oh, that's a great point, Chris.
Yeah.
Our leader, Hui Huakai, Team Sponge leader.
There's no bias
King
here.
Sponge.
No bias whatsoever.
Yeah, that's really cool.
One of our viewers says that Otis won the Fat Bear 20.
Oh, it was Otis? He's an old guy. He's really cool. He often just sits in the pools and just waits for the fish to come to him, and he'll put his face in and grab them that way.
Wow.
Yeah, they're super fans of Bear Week too, that know all the different bears and watch pretty consistently.
That's pretty fun.
You know, maybe we ought to have the fat sponge contest.
I know. I know.
Which was the biggest sponge of the cruise on which watch?
Oh, well, the tallest, the longest.
The widest.
The widest? Yeah, we could have the tallest coral. The tall coral contest.
The biggest ones we've seen, and this one's the polyopagons, right? Are those the biggest ones we've seen so far?
Yep, absolutely.
And what was that species of sponge that you guys saw that was as big as the minivan on the Okeanos cruise? Oh, look at Saphanophore. Is that what that is?
It's in the family Rosellidae, and we didn't know even what subfamily it was in. But since then we do, when I was able to do the spicule prep and then shared all the photos with a guy in Moscow, and Dr. Konstantin Tabachnik, who's at the Shirshov Institute in Moscow. He's one of the world's authorities, so I always send him spicule photos and get his opinion on stuff as well. So then he concluded that it was in the genus Lophocalyx, but it's a new species.
Wow. Yeah, that thing's a beast.
Yeah.
It's huge. Thousands of years old.
Who knows? It was massive.
Yeah. Was that environment like really thick, soupy water? Was the water very,
not really.
Not particularly?
No. Just, I don't know. He's just been real successful.
It's kind of like that bear, just like sitting there and getting fat.
Yeah, and he had sponges growing on him too.
Huh. So. They provided habitat. That's wild. Do sponges have any defense mechanisms?
Oh, I'm sure they do.
Uh-huh.
Sure they do.
They're spiky. They're spiky, yeah.
Yeah, your spicules, I mean, what's the point of the spicules? One of their defenses, maybe, nobody wants to eat them because they get a mouthful of fish bones, if you will. And not fish bones, but the equivalent. Glass.
Fiberglass.
Yeah. We suspect that there's some other defenses as well, possibly some chemical defenses. Yeah. One of the more unusual sponges that I've ever collected is what we call the bleeding sponge, and I think I already told that story. We took a big polyopogon, by the way, and we took a chunk of it, and it immediately started turning red, not only on the main sponge where we'd taken the piece from, but the piece itself.
Huh.
And by the time we got it up to the ship, it was completely ruby red. So,
what did that? What drove
I have no idea. I've never seen a sponge do that before or since.
Wow.
It's possibly of some kind of a chemical defense. I don't know.
Yeah. Huh. Well, it's a lot of energy. It's a lot of energy to like create chemical defenses. I mean, that's you got to conserve your energy down at these depths.
Well, some sponges have commensal hydroids on them. And it doesn't seem to bother the sponge at all, and so they're kind of have another species, an idarian, that's actually providing them with a defense, a stinging defense.
Yeah.
So
That seems to be a popular strategy down here, like those hermit crabs with the anemones on their backs. Those are my favorite.
Yeah.
They're so cool. So is a ROV pilot nightmare, like trying to get something into a collection box and it not working out and going on forever, Trevor Antonella?
Like missing?
I don't know, like literally like a nightmare. I'm talking about literal nightmare, like you wake up in the night and it's from you. We're trying to put something in a box and it just never worked out.
Oh, I think the nightmare is more involved entanglement.
Oh yeah?
If the sample doesn't go in the box, I just get frustrated. But that's fine.
I also
that's different
have nightmares.
than sheer terror of vehicles getting stuck in each other or under stuff.
Oh.
That's the scary part for me.
What do you mean under stuff? You were like ready to go caving yesterday.
Oh, I love it, yeah.
Okay, so, so you're a risk taker.
No, I'm not. I manage risk, I think, quite appropriately.
I'd like to bring up some news that just came right now. So it looks like the transit to the next dive site is shorter than expected. So Emil is going to go change the board, and
somebody else gets blue water.
They're going to put the ROV in at midnight. Mark's up, and he's aware of it.
Roger that.
Yep.
I just did a little jig and dance.
It's fine.
I don't get stuck with the 3-hour blue water.
I don't really mind as long as we get on the seamount, but I'm just concerned about the storm at the back end of our cruise and trying to get in as many dives as possible, regardless of the time. That's the main issue. So blue water, it's not my preferred time during the dive, but doesn't really matter. As long as we can get our 14 dives in before we got to bail, because right now we can't even do the makeup dive. There's quite a front coming in.
Oh, really?
That's going to impact the entire Hawaiian chain all the way down to the Big Island.
Really?
Yeah. So.
Wow.
So we are racing to beat that. That's where I'm at.
We've had some exceptionally good weather for this cruise, I'd say, for winter in Hawaii. I mean, this is the time of unpredictable winds and big swells.
How big a storm are we talking?
It's more like a front, but it's the wave. The sea conditions are going to make it undiveable all the way from here all the way down through most of the islands.
Oh, okay.
And then we had a backup plan for the last dive to dive down on Middle Bank, which is the first bank past Ni'ihau, and get out of it. That's what we first thought the front was. We thought that was going to be okay water down there, and now the new forecast is no, it's not going to be good. And then we were also talking about bailing and doing some mapping south of here, but now it looks like no. So I don't know what's going to happen. We're just going to have to watch it. But right now, all I care about is if we can get our 14 up that we planned here, then it's already a successful cruise, but I wouldn't mind it being kind of close to perfection if we can get that 14.
Yeah. Go for it. So are we going map? Where's the next seamounts for? Is south or east?
No,
of us?
it's east of us. Already mapped.
Okay.
All we got to do is get over there fast.
Nice.
Dropping in at midnight.
Yeah, it's like 33 nautical miles directly to the east.
Yeah, it's going to be tight, but
it's going to be tight.
Sounds like Neil's about as worried about getting everything in as I am, so he's going to push. Does that give you guys enough time to turn the ROV around and do it?
Yeah, 4 hours is a good enough time to turn the ROV around.
Okay.
Roger. I saw him typing, so I think he's up, probably for recovery. Yeah.
Kind of inked.
on our WhatsApp. Navmap plus Justin. Yeah.
So ROV guys, what's the strangest, have you had any strange hitchhikers come up with the ROV that weren't expected?
I think the funniest was I was working with a Falcon ROV, and which is a small, I don't know, 150 pound vehicle. And there was probably 30 fish stuck in it on a recovery one time.
Oh, really?
Tiny little, I don't know, anchovy-sized fish. You have to like take the whole top off and pull them out of all sorts of nooks and crannies. But yeah, there's a lot of fish.
Did the whole thing smell like fish? Like
No, we got out pretty quick before it could start stinking. But definitely, if you don't deal with it immediately, then actually that has been a thing too. Is you smell some weird thing on the ROV, and there's a fish that swam into some spot and couldn't get out. Was out of sight for, you know, a week or two.
I would be surprised because a lot of these just come charging at the lights. You know,
We've had fish caught on the thrusters too.
You
can actually feel the difference, then it doesn't handle as well, and the fish are just going for a bunch of centrifuge laps.
Do we get any other questions coming in from our viewing public?
Not really.
No.
They're asking, is there a picture of that huge sponge?
There is on the, if you look up, I think if you just type up giant sponge Okeanos, you should get a picture of it. Let me make sure that that's right. But it was on one of the capstone dives in Hawaii for the Okeanos expedition. Okeanos giant sponge Hawaii. How about that? Yes, it's the first thing that comes up. So if you type in Okeanos giant sponge Hawaii, you can see some there's some great news coverage as well as the ocean exploration site about that find. That was 2016 May, right around my birthday almost.
It seems like everybody knows it's kind of a quick notice for those on the midnight to 4 a.m. watch, but Andrea's bolting to bed right now.
Oh, yeah.
So, yeah.
All right.
And Emil's with her, and it was Emil's decision there, so he's sacrificing himself for the team. So that's good.
He's amazing.
Yeah. Emil, excuse me. What did I say, Emil? Sorry, Emil.
Yeah, definitely check out that page with the giant sponge. I got a really great video on the front page about it. It's just a mutant. It's huge.
You just don't get the sense until you see that overhead view next to the ROV and just realize how massive that thing was.
Incredible. That's what we're about in Papahanaumokuakea. We're about big records, numbers, most important, largest albatross colony in the
big colony,
large colony, lots of corals, highest density of cultural sites in the Hawaiian archipelago. We've got a lot of, and we're just plain large. 1.5 million square kilometers is a pretty big area, about as from Las Vegas to New Orleans. If you were to lay it on top of the United States, it's quite a vast piece of the Central Pacific, North Central Pacific.
One of our viewers is asking if you can take a piece of sponge and grow it in your own home.
No, not these guys. Unless you got some place in your home that has about 3,000 pounds per square inch of pressure and freezing cold temperatures and total darkness and is completely salt water. No, it's too bad because we learn a lot about the sponge if we actually could keep them alive in captivity, but it's just too difficult, at least for now.
So like a sponge taxonomy, so like the cilia cells, I mean, do they share food? Do each cell, is it like each cell is on its own in terms of collecting and eating food, or are they able to share food between some type of connected mechanism between the cells?
I don't know. I should know, but I don't know the answer to that. I'll have to look that up.
Mm-hmm. Yeah, we commented on it before, but there's another cool video for you to look up if you look up green dye sponge. There's a great video that somebody did some experiments to show how currents are filtered, move through a sponge. Green dye sponge video. Hopefully that doesn't drop anything ugly, but no, it's it's the first thing that comes up. Sponge dye Sea Studios. Very cool video of this iridescent green dye moving through a sponge. It really gives you an idea of how complex these organisms really are. And you know, when we talk about what is the outlet side? What is the hole that's typically the operculum? Not operculum.
Operculum.
Is it operculum?
Yeah, operculum.
Okay. Yeah, at the top of the sponge, there's often either large pores or some of them you can clearly see a kind of a central hole at the top. That's where the outlet typically is. And this video really shows you how that works, how it ingests water and moves it through its interior matrices on the inside of the sponge and through these, like, they pump the water through some of the cells have flagellae and they can move the water through the sponge and it goes out through those operculae. It's really cool. So what got you so interested in sponges? Why sponges?
So when I was working for the Hawaii Undersea Research Laboratory, one of my jobs was to try to identify all of the animals that were imaged on video there. And that's pretty good at the fish because I already had this bottom fish project and knew a lot of the, shall we say, shallower but deeper fish. Corals, we had coral people around, so we had a lot of help with that. And there was a NOAA guy, Bob Moffat, who was really good with arthropods, so he helped us a lot with that. So we had all of these people, but then the sponges were this giant hole, you know, in our database, and it was just, you know, big sponge, holy sponge, radar sponge, all of these things, and I just decided, you know, this is a real problem. We've got to start dealing with this. So I just started working with sponges and contacted Dr. Henry Reiswig and started working with him on sponges, and then after a while I just got pretty fascinated by them.
Yeah.
And they really are. Once you, well, it's kind of like anything. Once you get into it, you know, you find that there's a lot of fascinating aspects to just about any job or any field.
Yeah.
You know, and this is just something for me that I hadn't really thought about before, and then I got into them.
A viewer asked a while ago if there are enough scientists to process the samples that we are collecting or that exist around.
Well, there's enough scientists on any cruise like this to put them to bed, but then when they go to the museums have got such huge collections in some cases, like the Smithsonian, and I worry that there are not that many scientists around. You know, more classical taxonomy based on morphology rather than genetics is there's not as many people interested in that anymore, but there's still a tremendous need for it. So that's why I sometimes say that taxonomy is very slow, and it's partially because of that. There's very few experts in some of the different animal groups that we work with, and you gotta get in line for them to take a look at your specimens, because they got a whole lot of specimens getting sent to them from all corners of the world. So it's very challenging.
Would you say that the primary area of, I guess, using those samples would be taxonomy?
The what?
The primary area of science that we use to actually take benefits of those organisms is taxonomy, followed by, because I guess if you don't know what kind of organism you're looking at, how can you refer to it if you want to use it in other areas like?
Yeah, well, I think there's an awful lot of interest right now in understanding, you know, taxonomic relationships in general with all animals, but in particular for us, deep sea animals, and figuring out how to actually go about conducting good genetic analyses of some of these animals. I mean, for a while, sponges, they had a lot of trouble getting good genetic data out of sponges, and now that's becoming better and better. But you're right. I mean, we're calling this animal this thing, and we're not absolutely sure, and you want to be sure, and you also want to understand the whole process of evolution. You know, what sponges are related to what corals are related to which corals, etc., sea stars.
And I guess there will be an urgency in that kind of research because the human population keeps growing exponentially, and with that comes the exploitation of resources, especially in the ocean. And so I guess
I mean, there are certain animals that I think hold keys to understanding how life evolved on this planet, and probably quite a few of those are in the deep ocean. We find more ancient forms of animals, such as sharks, for example, or crinoids or something like that. So we find these in the deep sea, and so they're.
And really, how long it takes to recover an area, an ecosystem that is destroyed.
Yeah, in the deep sea, it's really long, much longer than on land.
So if we do destroy certain areas of the sea, those could not maybe never be recovered again.
Well, it might recover, but probably not in our lifetimes or maybe our children's lifetimes. So it's going to take a really long time. So, but it's on this planet, it's one of the last unexplored domains. And you know, you've heard this before. I've said it once before, I think, if not twice. But Earth is a water planet. 70% of the Earth's surface is in the deep sea. You know, only 30% is land or terrestrial. And I always sort of contend if we don't know very much about the deep sea, we don't even know the planet we're living on. We think we do because we're a little bit focused on ourselves and our own needs, but we really don't know the planet. If you don't know 70% of the environment of the planet. So anyway, let me step off my soapbox. Get back on the ground.
All right, we're going to begin a setup for recovery here. We're at 85 meters. That's just a few meters until we hand over.
Great, thank you. I think we'll sign off now and let them do their thing. So
okay, so to all of our viewers out there, thank you for watching us on nautiluslive.org and following us in our YouTube channel. Instagram, Facebook, and Twitter.
Just to let you know, we changed the dive schedule. For those of you still watching, we're actually going to do another dive in pretty close to 4 hours here, so stay tuned. And we'll talk to you later.
Aloha. Ahui-ho.
Aloha.
Wait, is it 4 hours or is it 6 hours?
4 hours.
4.
Is it 4 hours?
Midnight.
8 to
Yeah.
midnight.
Oh, midnight. Okay,
Yep.
that's good to know.
Yes. Yes, it is.
It's 50 meters.
Deck control, we're at 50 meters, full stop.
Roger that, understood. Control van, are you ready for recovery?
Control van is ready for recovery.
Roger that, understood. Control van's ready for recovery. Bridge, deck.
Deck bridge.
Bridge, are we clear for recovery?
Jessica, proceed, please.
Roger that, understood.
Jessica.
Coming up.
Proceed, please. Love it. All right, let's get this guy going. Oh yeah, this. And now we see if I can fight that current, 0.7 knots.
You're going to do it.
Yeah,
Full faith.
She'll go. I think I'm up 12. It's fine.
Can I secure the mezzo, Trevor?
Yeah, go ahead.
I'm just staring at buck cam. I want to see a shark.
I want to see a shark bite.
Go for it, yo.
You can kill butt light whenever you feel like it.
I'm waiting until 10 meters.
Roger.
Not a meter sooner.
Roger that. Oh, it's spicier at the top here. See how it goes. It'll be fine.
Should I even have him reduce thrust? Do we need full forward motion?
Reduced thrust is good, yeah.
Okay.
I think the theory behind that one is so that
It's for Argus.
Argus doesn't
yeah,
get
no, I
bashed,
just didn't know if it would screw you up if we lost some momentum moving forward.
It might, but that's
It's okay.
just the nature of it, yeah.
Okay.
Might actually make it easier.
Cool.
Because I'm not relying on laterals, I can actually drive. Yeah, that's going to be on the... Might not be a goal. I feel like I get fewer goals than everybody else. I wonder if it's a me thing.
Maybe you just don't feel the pressure, you know? You're too cool for your goals. Don't worry yourself with these things.
Such frivolity. You can have them hold or cut thrust now.
Okay.
Then I can actually make some headway.
Bridge nav, can we reduce thrust on the jet pump? Thank you.
Come on, you. The death grip on this thing. So maybe if I back down. What happens if I back down? Let's try that technique.
And what about a holding position? You still want us to do that?
Yes.
Okay, roger. Just wait till Argus is up.
Backing up is not great for video.
Bridge nav, you can increase thrust on the jet pump and please hold position.
Look at that tether winging out. I'm actually making headway versus water compared to how much that tether's winging out. That's all the current.
Oh yeah, I see it. Wow. Yeah, for real.
Real deal.
It's not lying. Not this time.
Not this time. See if I can tighten it up. Well, also getting it stuck on my light bar, that'd be ideal.
Getting it stuck on what?
The light bar. See what I can do.
Showed up again. I know.
Now we're good. Now the daisy chain's off, lift lines in hand, I can drift wherever. It's all fine.
That was fine.
It's all right.
Of course it was.
It tends to be.
Yeah, good for when it's
We got procedures in place for when it's not. Okay, yeah, nice. No wrap. Woohoo, doggy. That's much better. Shark.
Yes. It's there again in the distance, coming towards us.
Let's do this. That's worse. Sorry. Just trying to light up the sharks, man.
There he is.
That's pretty cool.
I don't have to do anything now. The current's just doing my work for me. Sweet. Autopilot. Nature's autopilot. Just put her in bypass, sure.